Welcome to Chapter 11 of your Human Anatomy and Physiology book by Marieb and Hoehn. It's titled Fundamentals of the Nervous System and Nervous Tissue. This is a very foundational chapter for the next 4 chapters we're going to cover when we talk about the central nervous system, the peripheral, the autonomic. This is really the nuts and bolts of how the neurons themselves work and how nervous tissue works. So I'm going to try not to get lost in the weeds because it's easy to do. But I also don't want to rush through this too quickly. So the nervous system, believe it or not, is a little bit complicated. It's really immensely complicated, and the more you learn about it, the more you realize you don't know. But that said, we've made an effort to simplify it in this lecture, and in terms of its function, we can simplify it quite a bit, actually. The nervous system really only does 3 things. It collects sensory input, it integrates that input, and then it responds with some type of motor output, potentially. An example of that might be you touch a hot stove. You don't know it's hot. So you have temperature and pain receptors in your hand and fingers that are activated by that extreme temperature, which results in sensory input. You have what's called action potentials or nerve impulses being sent to your spinal cord at a very rapid rate. Some integration occurs in your spinal cord, and as a result of that integration, you move your hand. You produce motor output, and motor output is in the form of action potentials being sent to your arm muscles so that you withdraw your hand. It's actually an example of a reflex as well. But that particular reflex illustrates the 3 functions of your nervous system. Now it can be much more complicated. Sensory input could be, well, you're listening to my voice right now, and that's reaching your brain in the form of sensory input, and you're integrating my words in your language association centers, and you're learning. And you may not be doing any motor output. You may be fighting to keep your eyes open. Maybe that's your motor output. So not all 3 things have to happen at once, but these are just the 3 functions that your nervous system can perform. Again, in terms of the anatomy, we do our best to simplify the nervous system as well. So the first way we can break your nervous system down is by categorizing the parts of the nervous system as being central or peripheral. So your brain and your spinal cord comprise the central nervous system. Everything else, everything outside of your brain and spinal cord, is a part of your peripheral nervous system. This is a human categorization. What I mean by that is anatomically, there's no line that says, okay, this is now peripheral. Your nervous system is one integrated system. But for the sake of learning how it works, it's very helpful to break it up into components. So this is just the first of many components we'll break the nervous system up to. We'll do autonomic, we'll do somatic, we'll do visceral, we'll do all kinds of things. And a part of that is right here on the slide. So this was an anatomical differentiation, central and peripheral. This is a functional differentiation. So we're saying that in addition to having 2 anatomical divisions, central and peripheral, we also have 2 functional divisions, sensory and motor, also called afferent and efferent. I prefer sensory and motor, so for obvious reasons. So sensory, I think you guys know what sensory means. It's a sensation. Some type of sensory receptor is being activated by some modality. It could be pain, temperature, vibration, pressure, light touch, deep touch. It's activating a sensor in your skin or your joint capsule or your muscle, and that's producing nerve impulses which are racing to your spinal cord and potentially your brain. So most of those examples, I think all of those examples I just gave, fall into the category of somatic sensory fibers. So these are things in your flesh, in your muscles, your bones, your joints, your skin. Pain, temperature, touch, light touch, all that stuff I just named, that's somatic sensory fibers. We also have visceral sensory fibers. So visceral refers to your viscera, so your organs. And if you've ever had a stomach ache, you've experienced stimulation of a visceral sensory fiber. I'm using this word fiber here. I could also be saying system, and this is arbitrary, why I switched to system down here. This is just referring to, I could be talking about a specific neuron fiber or a collection of all of those fibers, in which case I'd be calling it a system. I'm sorry if that's confusing. But anyway, so the point is you can have somatic sensory neurons and visceral sensory neurons. You can also have somatic motor neurons, neurons that move skeletal muscle. So if you want to lift a heavy weight, you're going to be recruiting somatic motor neurons. And we talked a bit about that in the muscle chapter last quarter. You can also move other types of muscle. So cardiac muscle, smooth muscle, glands. You don't move glands, but you can stimulate secretion from glands. That's all done by the autonomic nervous system. That is going to be its own chapter, so I won't dive too much into that, but it's an independent autonomic branch of our peripheral nervous system. It's a motor branch that innervates cardiac and smooth muscle and glands that helps us to maintain homeostasis. There's a lot of reflexes involved. If you've ever stood up and gotten lightheaded and then felt your heart beat harder to compensate, you've experienced an autonomic nervous system reflex. That's a fun chapter, so I'll save it for then. I like these check your understandings because I always put things in here that I sometimes forget to really stop and cover. So I used the word integration earlier. What is meant by integration? And does it primarily occur in the CNS or the peripheral nervous system? So this is prompting me to really take a deeper dive into what these two branches are. We know the CNS is the brain and spinal cord, and we know the peripheral nervous system is everything else. But what exactly does that mean? So the peripheral nervous system, everything outside of your brain and spinal cord, it's primarily axons. Now, the book explains axons after this section, which is unfortunate, but let me just say now that axons are really the part of the neuron which transmits electrical signals. There's another part of the neuron that collects them and integrates them. But I guess the point is that the peripheral nervous system, you can think of that like copper wiring. It's just transmitting an electrical signal. There's no complex decision making occurring. There's no central computer. It's just copper wiring carrying electricity. So that's really going to lend itself towards sensory input and motor output, bringing sensory information in and taking motor information out. The CNS is more analogous to that central computer. That's where decision making and processing occurs. Okay, so that's where you decide what to do about that burning sensation that you're experiencing when you touch a hot stove, and you decide to move your hand away. Actually, that decision is so subconscious, it's a reflex, but there's much more complex decisions you can make. Anyway, so the purpose of this question is just to illustrate that integration, that decision making that occurs between neurons primarily occurs in the CNS. A more scientific definition would be to say that it occurs anywhere that two neurons synapse with one another. But just kind of for this intro, just sort of basic conceptual understanding level, it's decision making, it's processing, it's integration, and that primarily occurs in the CNS. Okay, so quick little quiz here. Which subdivision of the peripheral nervous system is involved in relaying the feeling of a full stomach after a meal? So stomach, that would fall under viscera, so that would be sensory as well, because it's a feeling of a full stomach after a meal. That would be visceral sensory. How about contracting the muscles to lift your arm? That would be somatic, somatomotor, or somatic motor system, or a somatic motor fiber, if we're just talking about one neuron. Also, increasing your heart rate. Well, that's, you might say viscera, but um, visceral, that was pertaining to sensory specifically. This is a motor action. We're increasing your heart rate. So the branch of your nervous system that innervates cardiac muscle and smooth muscle and glands that could do something like this would be your autonomic division. And that's even the example I used earlier. When you're sitting on the couch, maybe you just ate a big, delicious Boca burger, um, and and the doorbell rings, and you jump up, and your vision starts to go white, and you start stumbling because your blood pressure is so low, because you were so relaxed, and then your heart starts to pound. That's the autonomic nervous system that does that. Okay, let's move on to nervous tissue, histology of nervous tissue in particular. So, nervous tissue is 80% cellular. Compared to connective tissue or something like that, that's pretty highly cellular. And there's only two cell types. There's multiple types of cells within this one category, but there's two general types of cells. So we have neurons and we have neuroglia, or glial cells. I've also heard this pronounced neuroglia, but I say neuroglia. Anyway, neurons and neuroglia. Neurons are outnumbered 10 to 1 by neuroglia. You can see neurons are much bigger and they're much more complex. And then you see all these little nuclei around the neuron. Those are neuroglial cells. Their job is to support the neuron. And we're going to actually talk about all 6 types right now. So there are 6 total types, 4 in the central nervous system, 2 in the peripheral. So let's look at central first. The first one we have here is called the astrocyte. That's a good name for it. It kind of looks like a star cell, so an astrocyte. The astrocyte, let me identify it here. It's in green. Excuse me, sorry, one second. Okay, so the astrocyte is in green, and you can see, I don't know if you can see my cursor in this recording or not, but it's hugging the capillary. So you have this green starfish cell hugging the capillary, and you also see that it's connected to the cell body of the neuron. This is the neuron right here. That's interesting. It tells us a little bit about what it might do. So, you know, one thing that's interesting about the brain is blood is toxic to your brain. And you might be thinking, well, that doesn't make any sense. A stroke, if you cut off blood supply to the brain, you have a stroke and it's devastating. So I'm not saying that your brain doesn't need oxygen and glucose, because it does, and a stroke will deprive it of that. But actual physical contact of blood onto a neuron is toxic. It's toxic to neurons, and also if there's a bleed in your brain, that's going to increase cranial pressure, and that's also very bad. Often the cause of death in a motor vehicle accident is that increased cranial pressure, and your brainstem actually can make contact with your foramen magnum, and that's not good. So anyway, sorry. So the point is here that you don't want blood on your neurons, but you need oxygen and glucose and other things from your blood. So that's a conundrum. So the answer to that conundrum is this cell called the astrocyte. The astrocyte is able to pull the required nutrients out of circulation. It's able to pull out the oxygen, the glucose, etc., out of the capillary and deliver it to the neuron cell body. Okay, and it's perfectly positioned to do that. And then it can remove waste from the neuron and return it to venous circulation, not in this picture. Also, it can just be released into the cerebrospinal fluid, which makes its way back to venous circulation anyway. So the astrocytes are responsible for maintaining the external environment of the neurons and making sure they get the appropriate and necessary nutrition and oxygen needed for them to function properly. There's a fancy word, it's not that fancy, for all of the astrocytes collectively, and that is the blood-brain barrier. the blood-brain barrier specifically is a barrier that any chemical must pass to reach a neuron. So it's going to need to travel through this capillary wall, which in the brain is very tight. In most parts, capillaries leak. In most parts of your body, they leak fluid. In the brain, they don't. They're stitched up by tight junctions pretty effectively. They're fairly impermeable. So they have to get through the capillary wall, and then they have to get through into the astrocyte, and they have to be transported to the neuron. So that's the blood-brain barrier. Microglial cells are here in this little light blue, not these funky looking ones down here, but up top, this little light blue microglial cells. They're kind of like the white blood cell of the central nervous system. Remember, blood doesn't enter the nervous tissue of your central nervous system, so there are no white blood cells. So you need some type of immune cell, and that's a microglial cell. It can detect and destroy pathogens. It can destroy non-functional tissue. It functions like a white blood cell, just outside of your blood. It's instead inside of the central nervous system. To left, we have ependymal cells. They're in pink here, lining, typically lining a ventricle or a fluid-filled compartment within your brain. They're ciliated, and the cilia moves and circulates cerebrospinal fluid. So their job is to move the fluid in the brain around. We don't really have a lot of ponds here in California, but if any of you guys grew up in the Midwest or the East Coast, there's a lot of ponds. I remember there was a nice neighborhood, not where I lived, but a ways down, that I had a friend who lived in. Oh, I'm trying to remember what it was called. I don't remember. Anyway, it was in Michigan, in a town called Clinton Township, and there was a big pond in this subdivision, is what we called them. Like, it's like a gated community, I guess. Anyway, so I remember there was a period of time where the fountain in the middle of the pond stopped working, and within a couple days or weeks, it got really green and gross, started to smell bad. It became stagnant. That's almost the idea here of these cells, is they're kind of like the fountain in that pond. They keep the fluid moving so that it can be recirculated, returned to venous circulation, so new fluid can be produced by a tissue called choroid plexus. That's next chapter. And it can keep that cerebrospinal fluid flowing. And then the last one here, it says a great name, oligodendrocytes. So oli or oligo is basically just means a few. Dendro is a branch or tree, I think it's a branch, and cyte is a cell. So this is a cell with a few branches. And if you look at the picture, that fits. So there it is in white. You can see that its arms are extending from its cell body and wrapping around axons of adjacent neurons. So axons specifically are the tail. So the tail of the neuron is called the axon. This is the part of the neuron that transmits nerve impulses or electrical signals. So what they do is they actually speed up the transmission of action potentials. And we'll explain how that works in a little bit. We have two neuroglia in the peripheral nervous system as well. They're called a satellite cell and a Schwann cell. These are basically just counterparts of glial cells we've already talked about. So a satellite cell, for example, is like an astrocyte, but for a peripheral nervous system neuron. So here, if you see the cell body of the neuron, is surrounded by satellite cells. So these cells protect it from exposure to blood. They maintain its extracellular environment. And then we have Schwann cells, which are myelinating. We didn't really talk about that yet, I'm sorry. But so oligodendrocytes, they were wrapping this fatty appendage around the neuron. That process is called myelination. And we have the same thing in peripheral neurons, except instead of using oligodendrocytes, we're using Schwann cells. So each one of these is an individual cell. Okay, so what type of glial cell controls the extracellular environments around neuron cell bodies in the central nervous system? That would be the oligodendrocyte. In the peripheral nervous system, that would be the Schwann cell. Which two types of neuroglia form insulating coverings? Oh, I'm sorry, guys. Did you catch my mistake? Maintain the extracellular environment. That would be the astrocyte in the CNS and the Schwann cell in the peripheral nervous system. Excuse me. So astrocyte. Oh no, I'm going to sneeze 3 times. Maybe 2. So astrocyte for central, Schwann for peripheral. Now, which two types of neuroglia form insulating coverings called myelin sheaths? So in the central nervous system, myelination is done by the oligodendrocyte. In the peripheral nervous system, myelination is done by the Schwann cell. All right. Neurons. So we're finally addressing the most important cell of the nervous system. This I feel like should be the first slide, but here we are. So the neuron is the structural unit of the nervous system. This is the functional unit of the nervous system as well. It is what collects electrical signals, integrates them, and then transmits action potentials, which are a special type of nerve impulse that we will talk about in the second part of this lecture. So anyway, these cells have 4 special characteristics. Most obviously, maybe, is their ability to conduct electrical signals called nerve impulses. Not many cells can do this. Muscle cells can transmit these same signals, but other than that, it's really just the neuron. And the neuron is exceptional at it in that it can have tens of thousands of connections with other neurons, all of which are able to send and receive electrical signals, which is what makes the nervous system so complicated, is this immense amount of interconnectivity between these cells. Another interesting feature, important characteristic, is that they have extreme longevity. Extreme longevity. Their longevity is about the same as your longevity. So they can live up to about 100 years, which is, one of the reasons why we live up to around 100 years, potentially. Not much longer than that. Neurons are amitotic. They cannot divide. So at first that sounds like a bad thing, and I guess it is. Because if you have a stroke or a spinal cord injury, it'd be nice if these cells could divide. And you'd be able to heal from that injury much more quickly. You get a cut in your skin, and the keratinocytes and the fibroblasts, they divide and they rebuild, and you heal quickly. You get an injury in your brain, that's not necessarily the case. Although your brain is pretty remarkable at recovering. It's pretty interesting to study. Anyway, the point is they're amitotic. Now, while that may sound like a design flaw, it isn't really, because these cells, they're not just chemical in nature. They have a structure to them. They form pathways with one another. Like I said, one neuron might be connected to 10,000, it's really much more than that, because the axon could be connected to 10,000 other neurons, but then the dendrites could be receiving impulses from any number of thousands of other neurons. So you form these pathways, and if you've ever learned something, you formed a pathway, right? You're learning how to walk as a baby. You're forming and reinforcing these pathways for walking. You learn to talk. You're developing pathways in your Broca's and Wernicke's area in your brain. So there's interconnections, and those interconnections are physical. So if one of those neurons was to divide, it would destroy all of those connections. And this is a silly analogy. I'm not even sure it makes sense. But if any of you have ever seen the Adam Sandler movie 50 First Dates, where she forgets everything the next day, this is to me an illustration of what would happen if your neurons divided every night. All of your memories would just evaporate. Although you wouldn't probably know how to speak or walk either, so it'd be a little bit more extreme. But tangent, I actually ran into Adam Sandler once at Mammoth Mountain Village, and I'm like, man, what is that who's this loud? I heard a loud guy. I'm like, that sounds like Adam Sandler. What the heck? And it was Adam Sandler. So I don't think he's an actor because he was his exact same like version of himself in movies and real life was my interpretation. So anyway, sorry for the tangent. In class, I'm much worse about tangents. I'm usually better on the recordings. So, okay, we're going to look at structure. This is an image of the cell body. So I guess all I'm really trying to illustrate with this photo here is that this is where all of the organelles are. So you can see the nucleus, the mitochondria, the endoplasmic reticulum, Golgi apparatus, everything is here. There's minimal organelles in the axon. Okay, this is a better picture anyway, so let's look at this. So here's the cell body, and you can see that there are processes radiating out from the cell body. This is a very typical, what's called a multipolar neuron. This would be the type of neuron, potentially, you're going to have millions of these in your brain, but this is kind of the standard neuron of the motor division, the somatic motor neuron. So if you decide to lift something heavy, a somatic motor neuron like this will be involved. So they don't all look like this is my point, but we're going to use this as our model for right now. So the first process is coming out of the cell body is called the dendrite. So the dendrites in this photo are these short, non-myelinated processes, and there are several coming out of the cell body. The axon is this singular, much larger myelinated process, and it has several regions. It has a hillock, which is this funnel between the cell body and the axon. It can have collateral branches coming out at right angles that are rather large. There's terminal branches, and then there's axon terminals, which are actually the very terminal ends of the neuron. And you can have tens of thousands of those on one neuron. 10,000 is kind of like a large amount, but you can have even more. Some neurons have less, some have more. So it's this pretty complex structure. You can see that mitosis is not going to work out very well for this type of cell. I don't know if I have another slide. Yes, this is what I'm looking for. So we're going to talk about the functional characteristics of these three regions. So the dendrites in green, they collect electrical signals. That's their job. So they collect what are called graded potentials. And so you may get one graded potential that's excitatory, which means that it is allowing positive ions to enter the cell. And you may get another graded potential that's inhibitory, which means it's allowing either negative ions in or positive ions out. There's a net negative response, I guess, is what I'm trying to say. So an excitatory synapse would make the inside of the cell more positive, and inhibitory synapse would make it more negative. Anyway, so you have all these electrical signals coalescing on the dendrites, and that resulting ion flow, whether positive or negative, kind of works its way down to the cell body. Because this is all filled with fluid, and these ions are dissolved in fluid, and they can diffuse, they can float. So the dendrites collect the signals. The cell body integrates all of those signals. So you have some positive, some negative. It all kind of coalesces and collects in the cell body, and that's the process of integration. If a certain critical threshold value is reached in the axon hillock, then the axon can create a brand new electrical signal called an action potential. And that action potential flows down the axon, down to other neurons, or muscle, or a gland, depending on what it's connected to. And that's basically how a neuron works. So dendrites collect, cell body integrates, axons propagate. They transmit new electrical signals. So anyway, the axon is the conducting region. It conducts signals, it propagates signals. Propagate's probably a better word. The axon hillock, the junction between the axon and the cell body, is the trigger zone. That's where we have these special voltage-gated channels that open in response to a certain threshold potential, electrical potential, in which case they open and an action potential is produced. If none of this is making sense, that's okay. We're going to take a lot more time to explain all of this. It's baby steps. When the action potential reaches the axon terminals, a chemical is released called a neurotransmitter. Last quarter we talked about acetylcholine a little bit. This quarter we'll talk about quite a few more. The cell membrane of the axon is called the axolemma. And when we're referring to the axon, we can even use the word nerve fiber. So you heard me say that. You heard me say fiber earlier. Fiber is referring to the axon. Axons can be quite long. You have axons that go from your L5 segment of your spinal cord in your low back, or really, I mean, L1, L2, even higher, all the way down to your foot, a single cell. So these axons can be quite long. They can be a meter long, give or take. All right, so I'm going to try now to edit in me going to another video talking about transport along the axon. If you don't see that video, it means I wasn't able to figure it out. But if you do see it, you'll be seeing it right now. Hey guys, so I'm not sure why there isn't sound, but that's okay, we don't really need it. We're gonna go zoom inside this guy's brain here. And they're showing these cool flashes of light representing action potentials. Of course, they don't really look like that, but that's okay. There's a cell body with lots of axons and dendrites converging. Sooner or later, I imagine, synapse. Sooner or later, I imagine we're going to see vesicular there we go. Okay, so these are vesicles filled with neurotransmitter being released from the axon terminal into the synaptic cleft, where they bind with chemically gated channels. But that's actually not what I really want you to see in this video. What I want you to see is how, remember earlier I showed you that all of the organelles were located in the cell body. So the organelles that produce neurotransmitter are all in the cell body, but that neurotransmitter needs to be released in the axon terminal, which is what we're seeing right now. So we're seeing microtubule highways being created, and they are created actually astonishingly fast like that. And these microtubule highways are going to be the track that our motor proteins use to transport vesicles filled with neurotransmitter. And there isn't necessarily anything in here that I'm going to quiz you about. I just think it's a pretty cool video, and it's a pretty cool idea. So any second now, I think we'll see some vesicular transport by motor proteins. Here we go. So there's dynein and kinesin. These are two different proteins that carry vesicles in opposite directions. Some carry proteins down the axon, some carry up, and they utilize ATP hydrolysis to change shape and drag vesicles filled with neurotransmitter. That neurotransmitter is produced by the ribosomes and the endoplasmic reticulum. It's modified there, and then it's carried one step at a time down the microtubule to the axon terminal, where it will eventually be released as a neurotransmitter into a synaptic cleft. So anyway, that's it for this video. Okay, and we're back. So hopefully that worked. So myelination. We're not going to talk a whole lot about myelination. I just want to reiterate that the oligodendrocytes myelinate axons in the central nervous system, and Schwann cells myelinate axons in the peripheral nervous system. In general, myelination accelerates action potential propagation. The signals propagate much more, they cover more distance in a lower amount of time. So the velocities increase. This is going to be important for things that preserve your life, like pain and movement. If you step on something dangerous, you want to be able to lift your foot away. If you see movement in the grass, could be a scorpion. You run. Oh, that reminded me of something. I just had some yard work done and I was told that I have scorpions in my backyard. I'm never going outside again. I'm just gonna probably just burn my house down because that's the scariest thing I've ever heard. So, anyway, sorry, tangent. Okay, so again, if I can figure out how to make these videos work, you will be seeing a video right now. All right, I haven't actually watched this video until this very moment, so hopefully it's good. Yeah, it looks good. So they're just showing a cross section, an animation obviously of a axon and how the Schwann cell wraps itself around the axon to myelinate a peripheral nervous system axon. The same thing is going to happen in the central nervous system. It's just done by oligodendrocytes instead of Schwann cells. So that's really it for that video. See you back at the slides. Okay, we're back. Which part of the neuron is its fiber? That would be the axon. How do nerve fibers differ from fibers of connective tissue and fibers in muscle tissue? Well, some of you guys weren't here last quarter, but when we say muscle fiber, we're referring to the entire muscle cell. So it's a little bit different because when we say nerve fiber, we're referring to the axon. In connective tissue, fibers are actual structural components. They're not cells at all. They're not even a part of a cell. They're found within the extracellular matrix. Now, we did not address nucleus. We're going to talk about that much more in chapter 12 when we get to central nervous system, but you will hear this term nucleus referring to collections of cell bodies within the brain. So if you see a collection of what we call gray matter is composed of cell bodies, dendrites, synapses within the brain. That will often be called a nucleus, like a basal nucleus. You might be more familiar with the basal ganglia. That's actually kind of an older way of saying it. So basal nuclei are nuclei within the brain, like the quadrate nucleus and the globus pallidus and the putamen. Anyway, that's all going to be next chapter. So don't worry about that right now. And I'm pretty sure we know the answer to this one. How is a myelin sheath formed in the central nervous system? It's formed by oligodendrocytes wrapping their processes around the axon, and its function is to increase action potential propagation speed. We're going to talk about why that is 2 chapters from now, I believe. Yep. So let's classify some neurons. And guys, we're actually almost done with part 1. So I may or may not get part 2 up today, but part 1 should give you something to work on while I get part 2 uploaded to anponline.com. So anyway, we only have a few slides left. So classification of neurons. Neurons are classified based on structure and function, like most things in anatomy. So neurons can be multipolar, bipolar, or unipolar. So this first example here is a bipolar neuron. It's called bipolar because it has 2 processes sticking out of it. Bipole. Okay. Let's go to the far right and look at. Oh, sorry guys. If I'm making myself yawn, I feel bad. I feel bad for you folks. Lab is going to be so much fun. Just wait. Anyway, so here's a multipolar neuron, and it's called multipolar because it has multiple processes extending from the cell body. We have multiple dendrites and 1 axon. Then we have what's called a unipolar. I'm sorry. This should say, oh no, unipolar. Some people call it unipolar, but more frequently it's called pseudo-unipolar, because it only has 1 process coming off of the cell body, but there's really 2 main processes, dendrites on one side, axon on the other. The cell body just happens to be in the middle. Okay. Bipolar neurons are fairly rare. They're found in your retina, for example. Pseudo-unipolar neurons are quite common. This is kind of the standard first order sensory neuron. So this is a neuron that brings information in from your periphery into the spinal cord. Multipolar neurons, these are kind of the standard motor neuron. So they take electrical impulses from your spinal cord out to your muscles. So just 3 examples, 3 structural classifications. Functional classifications, we have 3 as well. We have sensory neurons. This means that the impulse is carried from peripheral to central. It's carried in. Motor neurons are efferent, meaning they carry an impulse out, away from the central nervous system, out into the periphery. And then interneurons, these are neurons between 2 other neurons. So in this picture, the green neuron would be an interneuron. Most of the neurons of your brain and spinal cord are interneurons. Sensory and motor, these are actually in the periphery. They're bringing sensory information in, they're carrying motor information out. Okay, I like this question. So which structural and functional type of neuron is activated first when you burn your finger? So, I am so sorry, guys. I just made a few of you yawn. If you're curious, which I'm sure you're not, one of my circuit breakers blew out. So, oops, sorry about my dog. So I don't have air conditioning, and I actually don't have internet either. I'm going to have to put this on a USB, and I'm using my hotspot right now, but I'm going to have to put this on a USB and take it somewhere else, Starbucks or something, to upload it. So sorry for the delay, but so I don't have air conditioning or internet. I did not realize how much I like those 2 things, especially in the summer. So that's probably why I'm yawning. It's hot in here. But okay, so which structural and functional type of neuron is activated when you first burn your finger? So if you touch a hot stove, a pain receptor is going to be activated. Temperature as well, most likely, but pain for sure because it's a hot stove. So that pain is a sensory neuron, or pain receptors are attached to sensory neurons. So a sensory neuron is the functional type. But what's the structure of sensory neurons? The structure is pseudo-unipolar. So when you touch a hot stove, the first thing that's activated is a pseudo-unipolar sensory neuron. And then what is activated last to remove your finger from the source of heat? Well, that's going to be a multipolar motor neuron. So multipolar is structural, motor is functional. So multipolar motor neuron is what's activated to remove your finger. Okay, guys, this is where it starts to get a little heavy. Separate the men from the boys, I guess. The girls from the women. Oops, the women from the girls. It's a little complicated. Not so much here, but we're going to start getting into signaling. And signaling is when I sometimes lose people, so I apologize in advance. We're going to go through this fairly quickly. Part 2 is where most of the confusion happens. And you may find that some of this starts to make more sense after you see part 2. So let's just get through it together, and we'll shore up any weaknesses as we get closer to the test. So we're going to start talking about membrane potential. Now, the very first chapter of last quarter, we talked about resting membrane potential. So we're going to revisit that video on the next slide. But anyway, just to recap quickly, any membrane potential is just a separation of charge across the cell membrane. Think back to what potential energy is. If I put a rock up on a ladder, I've created potential energy because all I have to do is remove the ladder or push the rock and I create kinetic energy. The rock falls to the ground. It's the same idea with electrical potential. If I have a bunch of positively charged potassium ions and sodium ions outside of a cell membrane, and the inside of the cell membrane is negative, I have potential energy because all I have to do is open up a channel for those ions, and those ions will flow. They will go down their concentration gradients and down their electrical gradients, or both. So there's a potential for current, the flow of charged ions. Okay, so there's a few different types of channels. There's leakage channels. Leakage channels are always open, and they are very important in the establishment of resting membrane potential in particular, which we will look at. Then we have gated channels. Gated channels are gated either by a specific chemical. So if a specific chemical binds to them, they'll open. We talked about some of these last quarter. when we looked at acetylcholine-gated sodium channels, right? So these are specifically going to be involved in the formation of graded potentials. And then we have these guys, voltage-gated channels. These channels open at a specific voltage, say negative 55 is a common one. So at negative 55, these channels will open and then current flow ensues. And then we have mechanically gated channels. These are going to be more important in sensory receptors, like touch receptors and stuff like that. We won't be talking about those. But voltage gated and chemically gated, we're going to talk an awful lot about, because chemically gated channels are responsible for the formation of graded potentials, and voltage gated channels are responsible for action potential formation. So let's go ahead and look at this resting membrane potential video really quick. Okay, so let's take a look at resting membrane potential. I actually really like the narration of this video. I'm kind of bummed I can't get the sound to record, but it's okay. I'll do my best, so bear with me. So we're going to take a look at resting membrane potential. So in an unstimulated neuron, the inside of the cell membrane is about negative 70 millivolts more negative than the outside. So what he's showing you here is on the inside we have a lot of potassium and large negatively charged proteins. So the blue is the potassium, and that little chicken nugget looking thing is those are protein, large negatively charged proteins. Outside we have sodium and chloride. So I believe pink is sodium and yellow is chloride. Neither of these are particularly important. Right here there's no channels, so these ions are just bouncing off the wall. But once we introduce leakage channels, potassium is able to leave down its concentration gradient. So that's what we're seeing. We're seeing blue ions leaving because they're forced to, because they move down their concentration gradient. But as they leave, a negative charge starts to develop, because number one, these blue particles, it's potassium, it's positively charged. So as positively charged ions leave, the inside becomes more negative. Also, these large negative protein chicken nuggets are pulled closer to the cell membrane, and they're negative as well. So at some point the inside gets so negative that it starts to pull potassium back in. Ooh, my timing was good. All right, so now we're at equilibrium because we have a concentration gradient forcing it out, and we have an electrical gradient pulling it back in. That equilibrium occurs around negative 70 millivolts, and that's the resting membrane potential, negative 70 millivolts. So at rest, the inside of a neuron is a negative 70 millivolts more negative than the outer face of the cell membrane. And that is resting membrane potential. In an open channel, which factors determine which direction ions will move through that channel? So which direction ions will move through the channel? So that's going to be their chemical gradient or concentration gradient and their electrical gradient. So we sometimes call that the electrochemical gradient. For which cation, so that's a positively charged ion, is there the greatest amount of leakage across the plasma membrane? That is potassium. Potassium has the greatest amount of leakage. Membrane potentials that act as signals. So here's where it gets a little bit more confusing. We're going to focus the remainder of our energy on the end of this chapter, and then all of part 2 of this chapter, the end of part 1 and all of part 2, on comparing and contrasting graded and action potentials. We're going to explain how they're generated, propagated, how they're similar, how they're different, and how they are conducted. Here they are. These are it. There's two potentials that act as signals: graded potentials and action potentials. Here is a graphical representation of two different graded potentials. Okay? Check out the y-axis here for a second. We have negative 110 and 0 up top. So this graded potential this flat line represents resting membrane potential, negative 70. That's pretty common. Resting membrane potential. Then something happens. There's some sort of stimulus that causes it to move from negative 70 up to negative 55 maybe, if we're looking at the red line, or negative 60 if we're looking at the black line. Because we're moving from negative 70 towards 0, we're becoming less polar, right? Because 0 would be nonpolar. We're moving from negative 70 to, say, 60, we're becoming less polar. So that's called depolarization. And depolarization can take many sizes. A weak stimulus will cause mild depolarization. A strong stimulus will cause more significant depolarization. Both of these are graded potentials. That's where the name graded comes from, because they can be so many different sizes. They could even be different directions. So this would be an excitatory graded potential going towards 0. The graded potential could also go away from 0 and become more polar. That's hyperpolarization. You could have a weak inhibitory stimulus or a strong inhibitory stimulus. I'm saying inhibitory because we don't know this yet, but this is actually making an action potential less likely. That's why I say inhibitory. Whereas this type of graded potential makes an action potential more likely. I'm ahead of myself though, so don't worry about that right now. The point is that you can have depolarization or hyperpolarization of any different number of amplitudes. They're all graded potentials. So graded potentials vary in size and direction. That's why they're called graded, because there's many number of grades. Graded potentials continued here. Some kind of facts about graded potentials is that they are short-lived. Okay, so, and let me add to this, your book doesn't even really address it this early, but graded potentials are occurring in your dendrites of the neuron. And you can see this picture here. Hopefully you can see my cursor. They're zoomed in on a junction between an axon terminal and a cell body or a dendrite. That's where graded potentials occur. This particular graded potential is occurring at a synapse. So an action potential has traveled down this presynaptic neuron, and it's now triggering a graded potential to form in this postsynaptic neuron, and that's what we're looking at here. This is a great picture to illustrate what a graded potential is. Here's the axon terminal. It's triggering chemically gated sodium channels to open. Sodium is rushing in, and that is current flow. So we now have a positive current flow. Positive ions are rushing into this cell body right here. It's short-lived though, because as soon as that stimulus is removed, the channels close and the signal degrades. It's also local. The signal's strongest nearest the stimulus, because that's where the sodium is rushing in. So there's no sodium rushing in over here, so there's no signal. It's localized. Graded potentials are localized, and they're short-lived. And they decrease magnitude with distance. The further we get away from the entry point of sodium, the less sodium there's going to be. Okay? It's confusing when I'm talking about a chemical as a proxy for an electrical signal, but don't forget that these signals are charged. Okay, so the less sodium there is, also the less positive charge there's going to be. Anyway, so the point is they are short-lived, local, and they decrease magnitude with distance. We will find that none of this is true of action potentials. So that's where we're going to pick up part 2, is we're going to compare what we just learned about graded potentials with action potentials. And I imagine that graded potentials will start to make more sense once you have the big picture. All right, so I will see you in part 2.